The name Funaria is derived from the Latin word funis, which means a rope, and was used by Schreber in 1791 to denote the characteristic twisting of the seta when dry. As far as the systematic position of this genus is concerned, it belongs to division Bryophyta, class Bryopsida, order Funariales, and family Funariaceae. Funaria is the most widely distributed and common moss. It includes about 117 species which occur in almost all parts of the world. The genus is represented in India by about 18 species, which grow as dense green mats or patches in moist, shady soils, banks, rocks, bird grounds, and tree trunks. They prefer alkaline soil. Funaria hygrometrica is a very common species of this genus, growing extensively in the hills of various parts of the country. It is also called as Bonfire or Postfire species, as it is a pioneering agent which first appears in colonization at the burnt sites rich in ash and nutrient contents. The plant, as in other bryophytes, is the gametophyte. It has two distinct growth stages: a prostrate filamentous alga-like protonema and an upright gametophore. The protonema is a transitory structure, and the adult plant consists only of persistent gametophores or leafy shoots, which are 1 to 3 cm in height, that arise in numbers from a single weft of protonema. The conspicuous vegetative body of the genus is represented by these leafy gametophores that remain attached to the substratum with the help of rhizoids. It is about 13 mm or more in height. It bears flat, green, lateral expansions inserted spirally. For convenience, the central axis is called stem and the green expansions as leaves. Koch called the stem as colloids and the leaves as phylloids. The gametophores may be unbranched or show monopodial branching. The axis is covered with leaves which are spirally arranged in 3 by 8 phyllotaxy, or what we call octastachys. That is, in 3 complete spirals there are 8 leaves. The leaves crowd at the apex of the stem, forming bud-like head. Leaves are simple, sessile, ovate, and have a broad membranous base and a pointed apex. The mature leaves present at the base of the stem have a distinct midrib, but the younger leaves at the apex are devoid of midrib. Internally, a mature stem shows 3 distinct regions, which includes a central slender, cortex, and epidermis. The central slender is not differentiated into xylem and phloem, but consists of vertically elongated, narrow, thin-walled, colorless cells which lack protoplasm. These cells are called hydroids. Doyle in 1970 considered these thin-walled cells of the conducting strand or hydroids as functional counterparts of tracheids. The cortex is of relatively greater thickness in the young stems. Cortical cells are photosynthetic but lose their chloroplasts as the stem attains maturity. In the mature stem, the cortex is differentiated into 2 well-marked regions: thick-walled cells towards the exterior below the epidermis and the thin-walled cells surrounding the central slender. The cortex usually contains leaf traces which do not join the central conducting tissue, but end blindly in the cortex itself. On the other hand, the epidermis is the outermost layer composed of tangentially elongated chlorophyllous cells. It has no pores or stomata. In Funaria, water is conducted both externally by force of capillary between leaves and the stem, and internally by the central slender. Hence, Funaria is called as myxohydric moss. Internally, the leaf has a very simple structure. The lamina, also called as wing or blade, is composed of a single layer of large thin-walled cells which are rich in chloroplasts. The chloroplasts are peculiar in that they continue to divide even after the leaves have attained maturity. These chloroplast containing cells form the main photosynthetic tissue of the gametophyte. The midrib in the central part of the lamina consists of elongated cells, in which the leaves show a prominent thickening and provide mechanical support to the leaf. In the center of the midrib is a central strand of thin-walled cells. These probably aid in the transportation of products of photosynthesis to those parts of the plant where they are required. Stomata are lacking in leaf of Funaria. It takes place by the activity of a single pyramidal tetrahedral apical cell with 3 cutting faces. In this 3-faced apical cell, segments are demarcated regularly from each face. Each of the segments thus formed divide periclinally. The 3 inner daughter cells, by repeated longitudinal and transverse divisions, form the inner tissue of the stem. The outer daughter cells of the segment contribute the leaves, the subtending outer portion of the stem and the lateral branches. Branch primordia arise a few cells back from the growing tip of a stem. A superficial cell of the stem differentiates into a tetrahedral apical cell which forms the primordium. This cell is not axial in position but lies below a young leaf. It cuts off segments from its 3 cutting faces which forms the different tissues as in the main axis. Now, let us try to know something about reproduction of this genus. Funaria reproduces both vegetatively and sexually. Vegetative propagation in Funaria takes place by fragmentation of the primary protonema, formation of secondary protonema, gemma formation, bulbils, and apospory. As far as sexual reproduction is concerned, Funaria is monoecious, that is, male and female reproductive structures develop on different branches of the same plant. That is autoecious. Funaria is protandrous, which ensures cross fertilization. The female branches usually grow more vigorously and become longer than the male branches. As far as the position of Antheredium is concerned, Antheredia are born in groups at the expanded convex apex. They are surrounded by a rosette of bract-like spreading leaves called pericytium. These leaves are larger and are arranged more compactly on the axis than the vegetative leaves. A large number of long multicellular capitate hairs, the paraphyses, are intermingled with the Antheredia. As far as structure of this Antheredium is concerned, a mature Antheredium has a club-shaped body borne on a short massive stalk and an operculum at the apex. The body has a single layered outer jacket, the cells of which are polyhedral and flattened with numerous chloroplasts when young, but changing to orange or red at maturity. The operculum consists of 1 or 2 cells which are conspicuous by their large size, thicker walls, and more hyaline contents. Within the jacket, the body of the Antheredium consists of a dense central mass of numerous androcytes. As far as development of Antheredium is concerned, the Antheredium develops from a superficial cell, the antheridial initial. It is located at the apex of the antheridiophore. The antheridial initial becomes papillate and divides transversely to form an outer cell and a basal cell. The latter forms the embedded part of the stalk of the Antheredium. The outer cell undergoes 1 or more transverse divisions and forms 2 to 4 celled filament. The terminal cell of this filament divides by 2 vertical intersecting walls. Thus, an apical cell with 2 cutting faces is differentiated. The apical cell cuts off the segments in 2 rows in a regular alternate sequence. Now, upper 3 to 4 cells of this filament divide by diagonal vertical walls, each forming two unequal cells. The smaller peripheral daughter cell functions as first jacket initial. The large daughter cells again undergo similar divisions and give rise to second jacket initials on the outer side and primary antheridial cells on the inner side. Thus, each cell of the filament gives rise to two jacket initials and one primary antheridial cell. The jacket initials divide only anticlinally and form a single layered antheridial jacket. The apical cell of the filament forms operculum or cap region of the antheridial wall. The primary archegonial cells divide repeatedly, and the last generation of these cells form androcyte mother cells. The androcyte mother cells divide further and each forms two androcytes. The later differentiate into biflagellate anthrozoids. The development of the antheridia in an antheridial head is not simultaneous, and thus all stages in antheridial development may be traced in one head. As far as dehiscence of this antheridium is concerned, the antheridia dehisce when water in the form of rain or dew is collected in the cup formed by the closely arranged perigonial leaves. On absorption of water, the inner surface of the outer wall of the opercular cell becomes mucilaginous and swells. This results in the development of considerable pressure within the operculum. As a result, the distal end of the antheridium ruptures in the form of a pore through which the mass of androcytes wriggles out as slow stream of a viscous fluid containing the androcytes. The contraction of the antheridial wall and the development of the hydrostatic pressure of the antheridial contents also aid in discharge of anthrozoids. On reaching the surface of perigonial cup, the androcytes separate and spread out in the form of a thin film. The membranes of the androcytes dissolve and biflagellate anthrozoids are set free. They swim actively in water that accumulate in the perigonial cup. As far as position of archegonium is concerned, the archegonial branches arise laterally at the base of male branch. Archegonia are born in groups at the apex of these branches. They are less conspicuous since they are surrounded by a cluster of dull-colored leaves known as perichietial leaves. Paraphyses are also present intermingled with archegonia. Since the apical cell itself is used up in the formation of archegonium, the axis of the female branch has a limited growth. As far as the structure of archegonium is concerned, a mature archegonium has a long columnar stalk, a swollen venter, and an elongated neck. The wall of the venter is two-layered, but that of neck is single-layered, consisting of cells arranged in 6 rows. The neck encloses an axial row of 6 or more neck canal cells. The venter consists of venter canal cell and a basal larger egg cell. As far as development of archegonium is concerned, the archegonium develops from a superficial cell, the archegonial initial, which differentiates at the apex of archegonial branch. It divides transversely into a lower cell and an upper cell. The former divides repeatedly and forms the basal part of the archegonium, which is embedded in the tissue of the archegonial branch. The later functions as archegonial mother cell and divides by 2 intersecting oblique walls, forming an apical cell with 2 cutting faces. The apical cell cuts 4 to 8 segments on both sides alternately. These segments eventually form the stalk of the archegonium. The apical cell then divides by 3 intersecting oblique walls, forming 3 peripheral cells surrounding a central apical cell. The peripheral cells divide anticlinally into 2 each. The 6 jacket cells thus formed give rise to a single layered wall of the venter, which becomes double layered by further division. In Funaria, the neck develops from the derivatives of the axial cell and not by the divisions of jacket cell as in Hepaticopsida. The axial cell divides transversely into an outer primary cover cell and an inner central cell. The latter divides again by transverse wall into an outer primary neck canal cell and an inner primary venter. The primary neck canal cell undergoes transverse divisions to form a row of neck canal cells which occupy the lower and the median part of the neck. At this stage, the primary cover cell behaves as an apical cell. It has 4 cutting phases. The derivatives of the 3 lateral phases form the jacket of the neck, whereas those of the 4th basal phase forms a row of neck canal cells. The latter occupy the upper part of the neck. Thus, in Funaria, the neck canal cells have double origin. Those in the lower and median part of the neck are derived from primary neck canal cells, and those in the upper portion of the basal derivatives of the cover cell. A transverse division in the primary venter cell gives rise to venter canal cell and an egg. As far as dehiscence of this archegonium is concerned, in the mature archegonium ready for fertilization, the axial row of cells, neck canal cells and the venter canal cell, except the egg, degenerate to form a slimy fluid. The latter absorbs moisture and swells. As a result, the terminal cells of the archegonial neck are forced apart into a trumpet shape. During the process, some of the neck cells may get detached and are thrown off. A passageway down to the egg is thus formed. In Funaria, fertilization occurs as in other bryophytes. When the plants are wet with rain or dew, the apical cells of the Antheridium, which forms operculum, absorbs water and bursts. The spirally coiled biflagellate anthrozoids escape from the Antheridium while still enclosed within a vesicle. The vesicle eventually dissolves in water and anthrozoids are set free. Reaching the archegonial cluster, the anthrozoids swim to the archegonium. Entry of anthrozoids into the archegonium is due to chemotactic influence of the mucilaginous substances present in the neck. The chemotactic attraction is possible due to sugar present in the mucilaginous substance. Although a large number of anthrozoids enter into the neck of the archegonium, only one of them fuses with the egg nucleus. Union of male and female nuclei is complete within 10 hours. After fertilization, zygote secretes a wall, enlarges and divides to give rise to an embryo, which subsequently leads to formation of sporophyte. The diploid zygote formed by fusion of the anthrozoid with egg is the first cell of sporophytic generation. It secretes a wall around it, increases in size and enters upon active segmentation. The first division of zygote is transverse, forming an upper epibasal and a lower hypobasal cell. Both these cells divide by 2 oblique intersecting walls. As a result, a 2-sided apical cell is differentiated in the epibasal as well as in the hypobasal cell. The embryo at this stage has 2 growing points, each represented by an apical cell with 2 cutting faces. Derivatives of the apical cell gives rise to capsule and upper part of the seta, while those of the hypobasal apical cell gives rise to the remaining part of the seta and the foot. The process of fertilization also stimulates cells of the venter wall and they divide to form a protective covering called Calyptra. The Calyptra covers the capsule till maturity. This mature sporophyte of Funaria is much more complex than that of liverworts. It is differentiated into foot, seta and capsule. As far as foot is concerned, the foot forms the basal portion of the sporophyte. It is a small dagger-like conical structure embedded in the tissue of the tip of the leafy female branch and functions both as an anchorage and an absorption organ. The spatial role of foot in the absorption of nutrients can be deduced from the presence of wall ingrowths of food cells. The wall ingrowths in these cells increase their surface to volume ratio. Such cells in which surface area amplification is brought about by wall ingrowths are termed as transfer cells. As far as CETA is concerned, CETA is a long, slender but tough reddish brown stalk-like structure. It carries the capsule at its top and raises it more than an inch above the apex of leafy gametophore. It is differentiated into a central strand of small thin-walled cortex and epidermis with a cuticle. The central strand functions as a primitive type of vascular system. The thick-walled cortex gives strength to the slender CETA. It enables the CETA to bear the weight of the capsule. Conduction and support thus are the two functions of CETA. As far as capsule structure is concerned, the capsule is a highly organized structure. It is pear-shaped and green at first, but later turns yellow and then orange. Its upper portion is covered by a conical hood or a cap, the Calyptra. At maturity, the Calyptra falls off, laying bare its apex. The complex internal structure of the capsule can be studied in longitudinal section. It has three distinct regions. The apophysis, it is the solid, somewhat swollen, basal, green photosynthetic portion of the capsule. It also helps in conduction of water and food materials. It gradually narrows down into CETA, hence it is also called the neck of the capsule. Internally, apophysis is covered by epidermis. The epidermis is interrupted by stomata, which leads into air spaces below called the substomatal cavity. Another portion of capsule is theca proper. It is slightly bent middle portion of the capsule which lies between the apophysis and opercular region. It forms the fertile part of the capsule and can be further divided into following regions. For example, the columella. It is the central pith-like cylindrical portion of the theca composed of parenchyma cells. The columella provides water and nutrients required for the development of spores in the spore sac. The columella is surrounded externally by two elongated spore sacs. The outer wall of each spore sac is 3 to 4 layered, whereas the inner wall is only single layered. In young sporophytes, the spore sacs are packed with many spore mother cells. Each spore mother cell forms 4 haploid spores. However, letters are absent. A large air cavity is also present outside the spore sac. It is traversed by delicate filaments of elongated green parenchyma cells known as trabeculae. The trabeculae extend from the outer wall of the spore sac to the innermost layer of this capsule wall. The capsule wall is composed of many layers of thin-walled parenchyma cells. The outermost layer forms the epidermis. It is followed by a 2 to 3 layered hypodermis of well-organized colorless parenchyma cells. Next, there are 2 to 3 layers of chlorophyllous cells with intercellular spaces which constitute the photosynthetic tissue of capsule. The innermost layer of the capsule wall is connected with the outer wall of the spore sac through trabeculae. The apical region of the capsule consists of 2 important parts, the operculum and peristome. Operculum or lid, it is a conical obliquely placed cap-like portion of the capsule. Its 4 to 5 layers of cells in thickness. The inner 3 to 4 layers of cells are composed of small thin-walled parenchyma cells which are bounded by a surface layer which consists of cells with greatly thickened walls. It is the epidermis. The operculum at first is continuous with the theca region. Eventually, it becomes delimited by the appearance of a narrow circular constriction. Below the constriction are 2 to 3 layers of spatially radially elongated thick-walled cells. These form a circular rim or diaphragm. The peristome lies immediately below the operculum. It consists of 2 sets of long conical teeth, one within the other. There are 16 teeth in each set. The teeth of both the sets are on the same radii and thus opposed to each other. The peristome teeth are sculptured structures that close the opening of the spore sac. They are attached at the base of the rim or diaphragm. The outer set of the peristome teeth are hygroscopic and respond to the changes in the humidity. The presence of peristome teeth guarding the mouth of the theca region slows the rate of spore discharge. Above the rim of the theca, forming the broadest lower part of the operculum, is the annulus. It consists of 4 to 5 layers of cells. The upper 2 or 3 layers form a spatial ring of modified cells constituting the edge of the detached operculum. The lower 2 layers constitute the annulus proper. It consists of thin-walled cells with distended form. By the destruction of thin-walled annulus cells, the operculum is at first loosened and later dropped off. The mature capsule begins to dry up at the time of dehiscence. The thin-walled cells of the annulus break and the operculum is thrown away. Dropping of the operculum is assisted by outward hygroscopic movement of underlying peristome teeth. Due to this movement, slits between the inner thin-walled peristomal teeth become wider and spores escape gradually through these slits. In a wet atmosphere, the wet peristomal teeth bend inwards and thus closing the slit and prevent the escape of spores. In Funaria, there is a built-in mechanism which allows the dispersal of spores only when the conditions for germination of spores are favorable. The spores of Funaria are 12 to 20 microns in diameter and represent the first cells of gametophytic generation. The spore wall consists of 2 layers, the outer smooth and brown-colored exosporium and the inner colorless endosporium. The spore germinates under favorable conditions. It soon forms a long filamentous protonema. The protonema shows apical growth and forms 2 types of branches, the chloronemal branches and the rhizoidal branches. The chloronemal branches are characterized by the presence of chloroplasts in their cells. They are either prostrate, growing along the substratum, or may become upright. The rhizoidal branches, on the other hand, have oblique cross walls and grow downwards into the soil. The rhizoidal branches absorb water and minerals from the soil. The chloronemal branches give rise to many minute buds. The bud arises as a lateral swelling just behind a cross wall, near the base of the principal filament of aerial erect branch system, but occasionally they are formed directly on the brown filaments of the prostrate system. The apical cell cuts off 3 sets of lateral segments. The first formed derivatives usually do not form any leaf but contribute only to the base of the leafy gametophore. The rhizoids may also grow from this region even before the formation of the first leaf. A little later, the segments give rise to the leaves and the tissue of the system. The further growth of young gametophore is similar to that of apex of the mature gametophore. The mature capsule begins to dry up at the time of dehiscence. The thin-walled cells of the annulus break and the operculum is thrown away. Dropping of the operculum is assisted by outward hygroscopic movement of underlying peristome teeth. Due to this movement, slits between the inner thin-walled peristomal teeth become wider and spores escape gradually through these slits. In a wet atmosphere, the wet peristomal teeth bend inwards and thus closing the slit and prevent the escape of spores. In Funaria, there is a built-in mechanism which allows the dispersal of spores only when the conditions for germination of spores are favorable. The spores of Funaria are 12 to 20 microns in diameter and represent the first cells of gametophytic generation. The spore wall consists of two layers, the outer smooth and brown-colored exosporium and the inner colorless endosporium. The spore germinates under favorable conditions. It soon forms a long filamentous protonema. The protonema shows apical growth and forms two types of branches, the chloronemal branches and the rhizoidal branches. The chloronemal branches are characterized by the presence of chloroplasts in their cells. They are either prostrate, growing along the substratum, or may become upright. The rhizoidal branches, on the other hand, have oblique cross walls and grow downwards into the soil. The rhizoidal branches absorb water and minerals from the soil. The chloronemal branches give rise to many minute buds. The bud arises as a lateral swelling just behind a cross wall, near the base of the principal filament of aerial erect branch system, but occasionally they are formed directly on the brown filaments of the prostrate system. The apical cell cuts off three sets of lateral segments. The first formed derivatives usually do not form any leaf but contribute only to the base of the leafy gametophore. The rhizoids may also grow from this region even before the formation of the first leaf. A little later, the segments give rise to the leaves and the tissue of the system. The further growth of young gametophore is similar to that of apex of the mature gametophore.